Stem cells have been controversial for many years due to ethical concerns regarding the destruction of fetal tissue. Many of these concerns were made moot over a decade ago. It was then that the stem cell researcher Shinya Yamanaka pioneered a technique for transforming adult cells into stem cells. Yamanaka was awarded the Nobel Prize in 2012 for this innovation. Nevertheless, in my discussions with people who do not follow this field, I've noticed that many are unaware of this. They remain resistant to the idea of developing stem cell therapies on moral grounds. It's a sort of knee-jerk reaction for many of them. This makes it clear to me that there is a real need to clear up this misunderstanding. I do this in service to the channel's stated goals of increasing funding and public support for regenerative medicine research. By the way, if I sound like a robot, that's because I am. Well, my voice is anyway. I have cerebral palsy, which gives me a severe speech impediment. If I were to make this video using my real voice, you probably wouldn't understand. Thankfully, I have Murph to help me out. Murph is a state-of-the-art text-to-speech AI program. I just feed it my script, and it spits out a synthetic voiceover within seconds. Murph has many male and female voices to choose from, with different languages and accents to fit your region. If you think you'd like to use Murph in your own projects, follow the link in the description. In this video, we're going to be looking at induced pluripotent stem cells, or iPSCs, what they are, how they are made, and their relationship to other types of stem cells. We'll then end with a discussion of the various ways they are used to treat and cure medical conditions. Part 1. The natural stem cell types. Before we can discuss iPSCs, we need a basic understanding of what they're trying to emulate, namely, the natural stem cell types. Stem cells in general are defined by their ability to do two things: 1, to multiply, or make more cells, and 2, to differentiate, or transform into the many specific cell types that make up the body. Stem cells are contrasted with somatic cells, which are fully differentiated and cannot multiply. When it comes to how stem cells are categorized, most people are familiar with the classification scheme that is based on the stem cell's origin, or where they are harvested from. According to this scheme, there are two types of stem cells: embryonic stem cells and adult stem cells. Embryonic stem cells, as the name suggests, are stem cells that are derived from human embryos. More specifically, they are obtained from the inner mass of a blastocyst, which is a very early stage of embryonic development. Nowadays, embryonic stem cells are obtained from the unused products of in vitro fertilization. Embryonic stem cells are used in stem cell research because they are capable of becoming any cell type that is needed. Adult stem cells are stem cells that are derived from adult tissue, as well as from tissues found in the later stages of fetal development. Most adult stem cells are tissue-specific, meaning they are already partially differentiated and therefore only capable of becoming the same types of cells that reside in the part of the body where the stem cell is located. The primary purpose of adult stem cells is to repair damaged tissue. Whenever an injury occurs, the adult stem cells nearby are triggered into multiplying and differentiating in order to replace the lost somatic cells. In addition to this familiar scheme, stem cells can also be classified by their potency. Potency, in this context, refers to how many different cell types a stem cell can become. Embryonic stem cells have the highest potency, since they can transform into any cell type. Somatic cells, by contrast, have no potency. The difference between an embryonic stem cell and a somatic cell is not binary, however. During embryonic development, stem cells move between different stages. As they do, they gradually lose potency as they begin to take on their eventual somatic cell identity. This progressively narrows the range of cell types that they can become. This is typically a one-way process. A cell cannot naturally regain its stem cell status. It can only become even more differentiated until it becomes a fully fledged somatic cell. Traditionally, this principle has been visualized as the so-called Waddington landscape. This idea was introduced by the developmental biologist Conrad Hal Waddington. It was a tool for conceptualizing the embryonic development process. As a cell divides, it metaphorically rolls down the various hills in the landscape. As it does, it occasionally reaches a fork in its path. Thus, the cell moves from undifferentiated embryonic stem cell to more partially differentiated forms. Finally, it lands in one of the available somatic cell identities represented by the valleys at the bottom. At this point, the cell loses its ability to multiply, and is therefore said to be terminally differentiated. This is the stem cell's fate. The different levels of the Waddington landscape correspond to the different stages of embryonic development. In the potency classification scheme, stem cells are categorized by level, which corresponds to their potency. The categories are as follows. Pluripotent stem cells can give rise to all the different types of somatic cells that make up the human body. Embryonic stem cells are pluripotent stem cells. There actually is a category of stem cells which are even more potent than pluripotent stem cells. They are called totipotent stem cells. Totipotent stem cells can become pluripotent stem cells as well as eventually form the tissue that connects fetus and mother. This includes the placenta and the umbilical cord. Blastocysts are composed of an inner layer of pluripotent stem cells and an outer layer of totipotent stem cells. Below pluripotent stem cells are multipotent stem cells. These are stem cells that are capable of becoming the cells that make up multiple types of tissue. One type of multipotent stem cells is called a progenitor cell, which can give rise to multiple closely related cell types. You may have heard of mesenchymal stem cells. These are a particular type of adult stem cell that are multipotent. They are usually found in adipose tissue such as fat or bone marrow. In addition to being more potent than other types of adult stem cells, they are also easier to harvest. This makes them valuable for the purpose of regenerative medicine. Oligopotent stem cells reside in a specific tissue and are only capable of becoming the cell types that make up that tissue. Finally, after that, there are unipotent stem cells, which are only capable of becoming a single type of cell. When unipotent stem cells differentiate, they become somatic cells. Most adult stem cells are either oligopotent or unipotent. Prior to Yamanaka's work, regenerative medicine focused on embryonic stem cells because they were the only pluripotent stem cells available. Their ability to become any cell type meant that they held great potential for developing cures for many conditions. However, as stated in the intro, many objected to their use because harvesting them required the destruction of fetal tissue. What was needed, then, was an alternative source of pluripotent stem cells, or, ideally, a way of forcing somatic cells to revert back to a pluripotent state. It was exactly this problem that Shinya Yamanaka set out to tackle. Part 2. Yamanaka's work. In 1999, Shinya Yamanaka became an associate professor at the Nara Institute of Science and Technology, or NIST, in Japan. Having earned a PhD in pharmacology from Osaka City University 6 years prior, he had then held a postdoctoral position at the Gladstone Institute of Cardiovascular Diseases in San Francisco before moving back to his home country. At NIST, Yamanaka was put in charge of his own research laboratory. As the lab's director, part of his job was to set a long-term goal for its members to focus on. He decided that this goal would be to figure out how to reprogram somatic cells back into an embryonic-like state. He had been inspired by several preliminary experiments done by other research labs showing that it was possible in principle. Based on these experiments, Yamanaka had a hunch. He had his team identify genes that were active in mouse embryonic stem cells but inactive or underactive in somatic cells. His theory was that some of these genes were responsible for keeping embryonic stem cells in a pluripotent state. If so, reactivating those genes should revive pluripotency in a somatic cell. They eventually identified 24 such genes. In 2004, Yamanaka transferred to the Kyoto University to continue his work, this time as a full professor. There, he set his new lab to the task of evaluating each of the 24 candidates. The genes were activated in mouse somatic cells using an engineered retrovirus to force packets of protein called transcription factors into the cell. Transcription factors bind to specific genes in the DNA strand and turn those genes on. In way of testing whether they were successful, they used a technique that let them replace one gene with another. They selected a gene only active in embryonic stem cells and substituted one that causes insensitivity to a certain antibiotic. If the engineered cells became resistant to the same antibiotic, that would indicate that all of their pluripotency genes had been reactivated. When the 24 candidate genes were activated one at a time, the result was a failure. None of them succeeded in reviving the pluripotent state. When all 24 were stimulated at once, however, the cells became insensitive to the antibiotic, indicating the restoration of pluripotency. These cells were also similar to embryonic stem cells in terms of their behavior, physical features, and genetic expression patterns. When the cells were injected into live mice, they formed teratomas, or tumors made of many different kinds of cells. These results showed it must be some combination of candidate genes that was triggering pluripotency through a synergistic effect. Through further experiments, the lab was able to narrow the list of crucial genes from 24 to just 4. These are OCT3/4, SOX2, KLF4, and C-MyC. The genes are now sometimes called the OSKM factors, but they are more commonly referred to as the Yamanaka factors. The next couple of years saw a flurry of activity for the Yamanaka lab. In 2005, they successfully used the 4 genes that they had found to create mouse pluripotent stem cells. In 2006, they published a now classic research paper, Induction of Pluripotent Stem Cells from Mouse Embryonic and Adult Fibroblast Cultures by Defined Factors, in the prestigious scientific journal, Cell. Finally, in 2007, they successfully used their method to generate human pluripotent stem cells. 5 years later, Yamanaka was awarded the 2012 Nobel Prize in Physiology or Medicine for this scientific breakthrough. He absolutely deserved it, as he could arguably be called the father of modern-day regenerative medicine. I am a big fan of Yamanaka, not only because of the huge contribution he made to humanity, but also because of the impact that his work could eventually have on me as someone with permanent brain damage. Simply put, this man opened the door to the possibility of ending disability in this century. In Part 3, we'll go into greater detail about exactly how Yamanaka's method transforms somatic cells into pluripotent stem cells. Part 3. Finding the epigenetic reset button. The power of the Yamanaka factors comes down to epigenetics. Epigenetics is the study of how cells turn their genes on and off in response to environmental cues. We have already seen how turning certain dormant genes back on can restore pluripotency in a somatic cell. Epigenetics also is what makes the difference between one type of somatic cell and another. Every cell in your body, whether it is a brain cell, a lung cell, a muscle cell, or a bone cell, has the exact same DNA. It is the specific combination of genes that are active in a given cell that determines its characteristics and behavior. Cells have various mechanisms for turning genes off and on. The exact state of those mechanisms is known as the cell's epigenome. Early in embryonic development, the stem cells in the blastocyst pass chemical messages between each other as they differentiate. These messages affect the cell's epigenome by triggering it to produce transcription factors. The activity of genes associated with these transcription factors cause epigenetic changes within the cell, activating and deactivating other genes. What genes get turned on in any particular cell will depend on the combination of messages that cell is receiving. This is determined by nearby cells, and will, in turn, determine the messages secreted by that cell, and so on. In this way, the cells coordinate their efforts as they go about the business of building a new human being. On the Waddington landscape, the balls always move in one direction, rolling downhill. This is because these epigenetic changes are irreversible. Subsequent modifications can be made to the epigenome, but the original epigenetic marks, as they are called, are never removed. As the stem cells are differentiating, they are also multiplying. When a cell divides to make more cells, each daughter cell inherits the parent's epigenome as well as its genome. The epigenetic modifications that lead to differentiation are made in between rounds of cell division. This allows the epigenome to be built up over successive cell generations. In each generation, the cells gradually lose their potency as the genes associated with differentiation and multiplication are turned off. Meanwhile, they gradually gain their somatic cell identity as the genes associated with that identity are slowly turned on. There is, however, one notable exception to the rule that epigenetic marks are never erased. As we know, a zygote is created by the fusion of a sperm and egg cell. But, sperm and egg cells are both fully differentiated somatic cells. This means that the zygote should inherit both their epigenomes and thus not behave like a stem cell. Luckily, there is an ordinarily dormant group of genes inside each cell called a transcriptional regulatory network. When activated, this network conducts an epigenetic reprogramming process which allows the human life cycle to start all over again. When Yamanaka found his 4 factors, he had essentially stumbled upon a way of artificially triggering the same network. It was like finding an epigenetic reset button. When Yamanaka's factors are introduced to a somatic cell, the same epigenetic reprogramming process that occurs in embryogenesis is initiated. This process is comprised of 2 steps. The first step involves the removal of all of the cell's epigenetic information. This erases the cell's somatic identity and returns it to an undifferentiated state. In the case of embryogenesis, this information would have represented the combined epigenomes of the sperm and egg cells. However, the process works just as well with any epigenome. This is why Yamanaka's method can be used to revive pluripotency in any cell in the body. The second step is the reactivation of the pluripotency genes, genes which enable the cell to begin differentiating and multiplying. After this, a second network of genes called an autoregulatory loop is activated. This is basically a genetic feedback loop that maintains pluripotency, readying the cell to become whatever cell types are needed. The result of this is the creation of what is, for all intents and purposes, an artificial embryonic stem cell. Yamanaka named this new type of stem cell induced pluripotent stem cells, or iPSCs for short. In Part 4, we'll explore some of the uses of iPSCs. Part 4. Uses of pluripotent stem cells. The most obvious use of iPSCs is, of course, the development of stem cell therapies. Apart from being free from the ethical hang-ups that have held embryonic stem cells back, iPSCs have 2 additional advantages. First, since iPSCs can be generated from the patient's own cells, there is no risk of immune system rejection. Before iPSCs, people receiving stem cell treatments would have to be carefully matched with compatible donors. Second, since iPSCs are derived from adult patient cells, there's a lot more of them to work with than those obtained from microscopic blastocysts. Other use cases of iPSCs also come from the fact that they can be redifferentiated into any desired cell type. This is done by introducing them to various combinations of transcription factors via engineered virus. By coaxing small collections of iPSCs into becoming a single cell type, scientists create what are called organoids. These are basically artificial mini-organs that are grown in a petri dish. Organoids have two primary uses: disease modeling and drug testing. In disease modeling, cells from those with genetic illnesses are used to create organoids composed of the affected cell type. These organoids are said to recapitulate the disease, meaning that aspects of the disease are recreated in them. They can therefore be studied under controlled conditions in order to gain a deeper understanding of the disease's underlying mechanisms. The knowledge gained is then used to devise new treatment protocols and to guide future research. Closely related to disease modeling is drug screening. Disease model organoids can be used to test new drugs that might be effective in mitigating the illness. This has two distinct advantages over traditional drug screening methods. Firstly, since organoids can be created en masse, multiple drug candidates can be tested at once without risk to the patient. This makes drug screening by organoid much faster and cheaper than conventional methods. Secondly, since human organoids recapitulate human diseases, they can be a more reliable drug screening tool than animal models. Conclusion. It wouldn't be a stretch to say that iPSCs have quickly become a cornerstone of modern regenerative medicine, as well as ushered in a new era of medicine as a whole. When stem cells were discovered, it conjured up all sorts of hopeful notions of what might one day be possible. It was thought that they could be used to repair damaged tissue, cure untreatable conditions, and even grow replacement organs. However, progress on this front was hindered for many years by the controversy surrounding embryonic stem cells. Because this is an apolitical platform, I won't include my personal feelings regarding the use of embryonic stem cells. Rather, this video's purpose was to show those who do consider this problematic that such worries are now largely unnecessary. Frankly, they have also become counterproductive. As mentioned in the introduction, I have found that many lay people won't look into stem cell therapy. Many won't even consider using it to treat their own medical conditions, or support their loved ones in doing so. More to the point, many remain resistant to putting institutional resources towards the development of stem cell therapies. And it is all due to these outdated concerns. It's really a shame that so many remain unaware of iPSCs and the story of how Yamanaka made his breakthrough. Because of their work, many of the dreams about what might be possible with stem cells can now become reality. If more people supported this technology's development, maybe we could push through the remaining barriers and achieve those dreams that much sooner. It is my sincere hope, therefore, that this video has, in some small way, helped in the removal of this hurdle. Maybe, after watching it, at least a few who had been hostile to stem cell research will change their minds. Keep watching to learn more about stem cells and the ways that they are being used to improve people's lives. The same goes for the progress being made in related biotech fields. I hope to eventually start posting regularly by the start of next year. It's looking like the format is going to be 50-minute videos twice a month. This will include one monthly update and one topical video. Thanks for watching the video. If you got something out of it, then please consider giving it a like and subscribing to the channel. You can also follow me on any of these social media platforms. And, if you really like what I'm doing, you can support me on Patreon. The links are all in the description.